Hi everyone, we're going to review pages 300 to 305. Please let me know if you have any more questions about this. I'm just going to review the important parts from this section. Okay, so 300 to 305 talks about a molecule called RNA. RNA stands for ribonucleic acid. Right? It's almost exactly the same as DNA, except instead of having deoxyribose, right, which is how DNA got its name, deoxyribonucleic acid, the sugar in RNA is ribose. Okay, so I'm going to do a quick comparison between RNA and DNA. So DNA, the sugar in DNA is called deoxyribose. And in RNA, instead of having deoxyribose, our sugar here is ribose. Okay, DNA is double-stranded. Hopefully you guys remembered we talked about it having that double helix structure with two strings twisted around each other. RNA is a single strand. Okay, for DNA, your four bases, your four bases are adenine, thymine, guanine, and cytosine, while in RNA, your four bases are adenine, uracil, guanine, cytosine. So everywhere that you would expect to see thymine, you're going to instead see uracil. That's how this works. And I'll explain it when we go over transcription, which is turning DNA and copying DNA into an RNA strand. Okay, the last thing that you need to remember is that DNA is a very large molecule that contains many genes. Okay, and RNA is a copy of one gene that is found on DNA. Whatever it is that gene, or whichever gene it is that it needs to copy for whatever reason, to make whichever protein is necessary. Okay. Now, the whole purpose of DNA and RNA and genes in general is to make proteins, right? Genes are coded instructions in DNA. Whoops, wow, that's crooked. Okay. Genes are coded instructions for those who don't remember, found in your DNA that gives instructions on how to make proteins. Okay, because remember, proteins are the most important molecule in your body, right? They make up your enzymes, they make up a lot of different things and different parts. So to properly make these proteins is extremely important. Okay? Now, remember that DNA never leaves the nucleus, because if the DNA leaves the nucleus, the cell is going to take a chance on that DNA getting messed up somehow. right? If it's get used up too often, it might tear, right? If you're not careful with it, it might become mutated, and then the cell would be in trouble. So instead of using the DNA and the genes directly to make proteins, your cells are going to copy those genes into disposable units called RNA. Okay. So RNA is made to be disposable copies of genes that need to be made into proteins. Okay? Again, there's benefits to this, right? The first benefit to this is that it protects the DNA from damage, right? Because we're not actually moving the DNA out of the nucleus where it's safe and protected. It's not being used to make the thousands and thousands of proteins that your cells might need within a day. So it's going to protect that DNA, which is extremely important for the cell. Secondly, this allows the cell to make many copies of that gene. So if, let's say, it needs 1,000 enzymes of a particular type, it can make 1,000 RNA copies, and those 1,000 proteins, those 1,000 enzymes can be all made at once, which makes everything go a lot faster. Okay. Oops. What is wrong with my pen? Okay. So it allows it to make many copies of a particular protein all at once. That totally says time, by the way. Okay. So there are benefits to using RNA instead of using the genes and the DNA directly from inside of your nucleus. Okay. Now, the process of making RNA from your genes is called transcription. Okay. Transcription. Again, that is going from gene into RNA, right? Your genes are getting copied into a molecule of RNA. There's only one enzyme required for this. This single enzyme is called RNA polymerase. Okay, and RNA polymerase is pretty cool because it actually does the job of helicase and DNA polymerase all at one time, right? Remember from when we talked about DNA replication, when we need to copy DNA, in order to get to that DNA, all of that DNA has to be untwisted and unwound before your DNA polymerase can come in and start to make the complementary strand, the other half of that DNA molecule. Well, with transcription, RNA polymerase is going to do the job of both at the same time. So what's going to happen is you're going to have a strand of DNA. So let's pretend that this is your DNA strand. Okay. Whatever this strand is going to be. I don't know. I'm just going to use 2 colors. Wow, those are ugly together, but that's okay. Doesn't matter. Right, so here's your DNA strand. You've got your purple strand, you've got your yellow strand. These are your bases holding them together. Okay. The enzyme RNA polymerase, let's use a different color for this. Okay, so RNA polymerase is going to come in and it's going to find the gene that it needs to copy. Okay, so let's say the gene is right here. Okay, that is the gene that RNA polymerase has to copy. And it's going to know that it needs to copy that gene because at a part or at a point here on that gene, you're going to have a little section here called the promoter. Okay, the promoter is basically like a signal to tell the RNA polymerase that that's where it needs to bind first, right? So this little RNA polymerase is going to come in, it's going to look for that promoter, and it's going to bind to where that promoter is signaling, right? Once RNA polymerase does that, it can then go straight down the DNA molecule, untwist it, and then make a copy of that RNA. Okay, so what happens next? You're going to have the next part, your DNA is going to get untwisted by this RNA polymerase right where that promoter starts, right? And again, what it's going to do is it's going to bind, it's going to bind to the DNA and it's going to untwist that DNA. And what that promoter, or sorry, not promoter, that RNA polymerase is going to do as it's untwisting that DNA is it's going to pick one of the DNA strands. And it doesn't matter which one because remember, they're exactly the same, just going in opposite directions. And it's going to start to make a copy of that DNA molecule, right, of that gene by base pairing. Okay, that RNA molecule is not going to stick to the DNA, right? It's going to be its own separate strand. So as the polymerase, RNA polymerase, moves along and reads that DNA strand, it's going to base pair. Wherever DNA has thymine, it's going to be adenine on RNA. Whenever there's guanine, it's going to be cytosine. And wherever there's cytosine, there's going to be guanine. However, because RNA does not have the base thymine, anywhere where DNA has an adenine, it's going to instead get turned into uracil on the opposite side. Okay, now I know that's a little confusing for some of you guys, but I'm going to draw this out. So let's pretend that our DNA gene has the following sequence on the yellow strand, right? Let's say it's G, C, T, A, T, C, C, G, A. Okay? So let's pretend this is the gene that is on the yellow strand of DNA being copied. When your RNA is being made by polymerase, every time you see a G on DNA, it's going to bind with C. Same thing with G, right? All of the binding is the same. However, when you see adenine, normally you would put thymine there, but since RNA is not made with thymine, you're going to have uracil there instead. Thymine still binds with adenine, right? All that other stuff, nothing changes. It's just wherever you would expect to see your thymine, you would instead see uracil. Okay? So that's transcription. It's pretty simple so far. RNA, remember, is its own strand. Once RNA polymerase is done copying that DNA, everything just kind of twists back on its own. That RNA molecule is then free to continue on to the next part of the process of transcription. Okay. So once it's done, the RNA molecule is made, basically, once the base pairs have been attached and stuff like that to each other, and this strand of RNA is made, you're going to need to edit your RNA molecule before it's ready to be used. Okay? So RNA must be edited before it can be used. Okay. There are sections of the RNA called introns that need to be removed and recycled. And then there are regions called exons that are kept in the final molecule. Okay. The reason why introns get removed is because they don't actually code for anything in a protein. They're just kind of extra, right? Why are they there? Some scientists believe that maybe they protect us against mutations, or perhaps they allow one gene to make many different proteins. We're not exactly sure quite yet. The exons will stay in the final molecule. Okay, so what does this look like? Well, let's pretend that we have our RNA molecule, C, G, A, U, U, A, G, G, C, U, and then we're going to continue this on. Let's pretend the rest of it, right, looks like, wow, my pen is really messed up. Okay, so let's pretend that this is the entire RNA sequence. Okay. Let's make exons a different color here so that we can kind of do this slightly differently. Okay, so let's pretend in this RNA molecule you have regions that need to stay in the final molecule, and then you have regions that don't code for anything, right? So during RNA editing, what's going to happen is you're going to have your RNA cut into pieces, right? It gets cut between every intron and exon sequence that there are. Okay. The introns are going to get cut out. Okay. So this is your intron. Intron is going to get cut out and removed. Why didn't it capture the G? Oh well. Okay. It's going to get cut out and removed. Nope, that's not it. I'm not sure why it's not working today. Because technology loves me. Technology loves me. All right. Not all right. Whatever. I totally know what I'm doing, guys. right, we're just going to undo that. Why is my pen just not cooperating with me today? Okay, well, you get the idea. These things are going to get cut out, and the final RNA molecule will not contain any introns at all. Okay, those introns are just going to get recycled and reused elsewhere, but they don't end up in the final molecule. Okay, so once all is said and done, this is your RNA molecule. This molecule is ready to leave the nucleus and head out into the cytoplasm for the next part of making proteins. Once the RNA has left the cytoplasm, it's going to find a ribosome. Or sorry, once RNA has left the nucleus and into the cytoplasm, it's going to go and look for a ribosome. Right? Because remember, transcription takes place in the nucleus because we don't want to remove the DNA from the nucleus. Okay? The reason why RNA is going to try to find a ribosome is so that it can be turned into protein. That code that it has on there is basically instructions on how to put together this specific protein. Okay. Okay. And this process of going from RNA to protein is called translation. Whoa, what in the... What? Why are you being such a butt? Ah! What? I don't know what's going on. Okay, well, ignore all of that mess right there. Okay, anyway, so translation going from RNA to proteins. What you're going to have is your 3 types of RNA working together in order to make this work. Okay, you have your messenger RNA, otherwise known as mRNA, which has the instructions. Your ribosomal RNA, which, like its name suggests, is found on the ribosome, right? It's also called rRNA. This is going to be the ones that read the instructions. And it's going to assemble the protein. It's going to put the protein together. Okay. And then lastly, you have your transfer. Oh my goodness. What? Why? What is going on with you today? Oh my gosh. Okay. And then we have our transfer RNA, otherwise known as tRNA. And it brings the materials to build the proteins. And the materials that build the protein are called amino acid. Okay, so it's going to bring amino acids over. Okay. So let's look at how translation works. All right, so what's going to happen is your messenger RNA is going to find a ribosome where the ribosomal RNA is going to start reading those instructions on mRNA. Okay, so step 1, messenger RNA is going to attach to a ribosome and your ribosomal RNA will read the code instructions, right? Same difference. On your messenger RNA. Now, those codes, instructions, they have a name. They are called codons. I know what it sounds like, but that's not what it is. Codons. Okay? Codons are read as every 3 base pairs. So what in the heck does that mean? That means that if I had this as my DNA strand, if I had this as my RNA strand, every 3 letters is a codon. That is one codon. That is your second codon. Okay? That is another codon. And this would be the last codon. Right? So every 3 letters is a codon. Okay? Codons, again, are found on your messenger RNA, your mRNA. Each codon is going to correspond with some amino acid. And there's like 20 amino acids. No, I don't expect you to memorize all 20 amino acid. Don't be crazy. I mean, you could. I don't recommend it. Okay. How do I know, or how does the ribosome know which codons go with which amino acid? Well, this is where let's see. Oh, I might not be able to input this picture. Oh, duh. Library image. I want to say it's here. Okay, so your ribosome is going to know which amino acid it needs, right? That transfer RNA needs to bring by looking at these codons, okay? You should be able to use this chart. It's going to be expected of you at some point. And this chart is pretty easy to use if you understand where to start. You're going to start basically in the middle, okay? The first letter of your codon is going to come from here. Okay, the first letter is in the lightest color. The second letter is going to correspond with this medium colored yellow. And then the third letter is going to be basically in the darkest yellow. Okay, and that's going to correspond with one of the amino acids that you see on the outside. So let's look at our RNA code, right? Our first codon reads CGA. Okay, now what we're going to do, let me just write these out real quick. UUC, GA, and then UCC. Okay, so what you're going to do is you're going to start in the middle. You're going to find the first letter. So in this case, it's going to be the letter C, right? Once you've found that first letter, you're going to be bound into basically this corner where the C is. Okay, you're not going to go C and then G and then A and be like, I don't know where everything goes. No, right? Once you find the first letter, you're going to be staying in the corner where that letter is found. So this C is going to be one of these 5 amino acids. Okay, so our first letter is C, right? Our second letter is G. So we are going to look at, whoa, what is that? Of course it is, of course it's going to screw up on me. Right, our second letter is G. That means that we're going to go to, why does it keep doing that? You know, some days are better than others, right? The second letter is G, which means we're going to go into, in that little corner, the next biggest letter. And the last one is A, and it tells us basically that the amino acid we need is arginine. Okay. Next one is UC, which means that our second amino acid is phenylalanine. Okay. Our third codon is GGA, so GGA. That gives us glycine, right? Notice I stay within the corner where my first letter is found. And then my last codon is UCC, so UCC, that gives me serine. Okay, so that's how ribosomal RNA is going to read the code on messenger RNA, right? So the first step of translation is for messenger RNA to attach itself to a ribosome. That ribosome has ribosomal RNA, which will start to read that genetic code, those codons. When it reads the codons, it's going to be able to tell which amino acid is needed in order to kind of make the protein. Okay. Now there is a special amino acid or special codon, AUG. I like to remember is AUGO, right? Is going to be your start codon. And it always codes for one amino acid, and that amino acid is called methionine. Every protein starts off with methionine because that's the start codon. It tells the ribosome to start here. The other special codons are called stop codons. Oops. They do not have any amino acids associated with them. They just tell the ribosome that this is where translation stops. Like, I'm not going to continue to make the protein. I'm just going to, finish the protein here. Okay. The stop codons can be found on here. Okay, wherever it says stop. There's 3 stop codons, UAA, UAG, and UGA. Now, I'm always going to provide you with the amino acid chart whenever we take a quiz or a test. If I expect you guys to translate the codons, I'm always going to expect you to or I'm sorry, not expect you, but I will always provide for you a amino acid chart, because again, you're not expected to memorize that stuff. Okay, the stop codons, the importance of them, they stop translation. Basically, again, tells the ribosome this is where the protein ends. Please stop adding more amino acid. Right? Doesn't matter how much is behind the stop codon. If you get to a stop codon, it stops. Okay? Same thing with the start codon. Doesn't matter how much stuff is in front of the start codon, the ribosome will not start until it finds a start codon. Okay? Hopefully that makes sense. So that's the first step. The second step is that transfer RNA brings amino acids over. To the ribosome. Okay. And it's going to help put those amino acids in the correct order. Let's erase that period. And it does this by matching up a section of the transfer RNA called the anticodon. Okay, the anticodon to the codon. Okay, so what in the heck does this mean, Tag? Right, so here's what this looks like. Here is your ribosome, let's pretend. Okay. Right? The ribosome kind of reads your messenger RNA code. Okay? Whatever that code is going to read is going to say, the ribosome, along with its ribosomal RNA, is going to read the code. And what's going to happen is right? It's going to, as it's reading the code, your transfer RNA, the little kind of messenger peoples, each transfer RNA is going to carry I have no idea what it looks like. I guess a clover? I don't know. Whatever. we're going to draw that slightly differently. It's going to carry its own amino acid, okay? The one that has AUG is going to carry the amino acid methionine on it, okay? And this transfer RNA is going to know where to put the methionine because it has an anticodon on it that is complementary, meaning what the other half would look like, right? A complementary sequence on it called the anticodon. That matches up. Why is it being such a butt? Oh well, whatever. That matches up with your codons, right? So this AUG for methionine is going to match up with the UAC, the complementary here, so that this tRNA knows that's where methionine goes, right? The next one, CCU, is going to have a transfer RNA that is GAA to match up with its 3 base pairs. Okay. And the CUU, if we look at our amino acid chart, right, CUU is going to be for leucine, okay? Leucine. So these transfer RNAs are going to bring your amino acids over to the ribosome, and it's going to start to put them together, okay? So the ribosome will put these amino acids together. So step 3, the ribosome will form what are called peptide bonds between the amino acids to form a long protein chain. We call this a poly for many, peptide for the bond, chain. And that's what a protein is. A protein is going to be a very large polypeptide chain. Okay? So at the end of the day, you're going to have something like this, where your DNA, A, T, C, T, G, A, C, C. Why do I keep doing that? A, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, so at the end of the day, if your DNA reads that, okay, your RNA translates it to U, A, U, C. Oh, I screwed up. My bad. And then I do it again. Because I'm a genius. Okay, U, G, G, U, G, U, A, G, G, right? So this would be your RNA code. Your protein would look like this, right? It's going to read this RNA code in codons. It's going to search for AUG as the start codon. So we're going to start off with methionine. Okay. And then the second amino acid, let me pull up my amino acid chart real quick on here. Hold on. Okay, so that's the first one. Our second codon is GGA. You could probably do this faster because you probably have the amino acid chart somewhere, unlike me. So GGA is going to be glycine. Okay. And then CUG is the third codon, and CUG is going to be leucine. Right? And then GUG is the next one. GUG is going to give us valine. And then UAG is the last code, right? UAG is a stop codon, so that's our amino acid chain, okay? Or that's our polypeptide chain, and that would be what our protein would look like if we were to transcribe and translate this particular DNA sequence. Okie dokie. Does anybody have any questions? If you do, please let me know and email me or something like that so that I can try to answer your questions as best I can.